So in the last video, we talked about the basics of linked genes, and I ended the video sort of discussing how we can tell what the chromosomes looked like beforehand, and we'll get to that in a separate bit. But in fact, I was sort of lying to you when I showed you the linked gene data. We don't usually just get the two parental phenotypes. We actually usually get data that looks like this. We do get all four categories. However, unlike in our old scenario with the unlinked gene, right? And remember this cross. Just want to keep in mind what the parents were here. If we were to do this cross with unlinked genes, we would expect 25% of each of the categories, and out of 400 total, it would be 100 of each. However, that's not what we're seeing here. We see these two large categories and these two small categories. The two large categories happen to be our parental phenotypes, and the two small categories happen to be what we call the non-parental phenotypes, the ones that don't look like the parents. What happened to create these options? Crossing over happened. You might remember that crossing over occurs during prophase 1 of meiosis. So we have our linked chromosomes. They happen to cross over, and then we get sort of a separate scenario. Again, the genes are linked, but they're now linked in a different combination. And so when presented with this data, you should know that this data, where there's two that are really large and two that are really small, that is indicative of linked genes. And you should also know that the two that are really large are going to be indicative of the parental phenotypes, and the two that are really small are indicative of the phenotypes that occur because of crossing over. So here I color-coded them. We can see the red ones with the high number of offspring are what the parents normally have, and therefore what they passed on normally. And then the orange ones with the low number of offspring are the result of crossing over. So those are linked genes.